Hello, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by Illyri Bio. It is my pleasure to introduce today's moderator, Dr. David Good. David is Senior Director of Business Development at Illyri Bio. After receiving his PhD in bioanalytical chemistry and spending nearly 2 decades in both lab-based and commercial roles, David has developed extensive knowledge of the drug development process. This includes providing bioanalytical, biomarker, DMPK, and spatialomics support for a range of modalities, from NCEs to biologics such as MABs, oligos, RNA therapeutics, and ADCs, ARCs. David Good has spent the last 11 years supporting pharma and biotech clients as they navigate discovery, preclinical, and clinical phases. Dr. Good, the floor is yours.
Thank you so much, Stacy, and thank you to everyone who took the time to join today. Good morning, good afternoon, and good evening. It's really my pleasure to discuss the CyTOF with you today. I thought we would start off just really quickly with a little brief introduction to who Illyri is. Illyri is new in name, but not in terms of our longevity within the industry. We actually have over 30 years in business providing bioanalytical and spatial omics support. We are about 160 employees, and in that time of 30 plus years, we have essentially seen most everything. You can never say you've seen it all because it's always changing, but we've actually worked with over 2,500 different drugs and metabolites, essentially every biological matrix that you can imagine, everything from the standard fluids to a range of different tissues. We are fully regulatorily compliant. We have done 16 audits with the FDA. And in that time, importantly, we have also analyzed over 5 million samples. So we support everything from early phase discovery R&D projects to preclinical IND enabling work, and all the way through phase 1 through phase 4 clinical. So we do have the capability to take on larger clinical trials as well as perform post-marketing surveillance. Next slide, please. So where are we located? We are actually a global company. We have sites. Our global headquarters is currently in Colorado Springs, Colorado. We also have a site in Salt Lake City. And most of the work that will be presented today is being supported out of our site in Lille, France. And so if you look down to the lower corner of our slide here, you may recognize our legacy brands, where Colorado Springs used to be a Pixent site. Salt Lake City was part of Tandem Biolabs. And then our Lille site, again, where Corinne is based and where this work will be performed, used to be EMA Biotech. Next slide, please. So with that, it's really my absolute pleasure to introduce you to Dr. Corinne Ramos. This is just a quick snapshot of her biography, but really, I don't know what the woman has not done. She has spent time in everything that you can imagine, starting at the NIH and the NCI, being in academia as a professor, transitioning into large pharma and being in the large pharma space before then ultimately moving on to supporting clients within CROs. So she has actually touched everything, essentially every aspect of drug development from academia through large pharma. So she definitely can understand your needs. With that, it's my pleasure, Corinne, if you want to take over.
Yes, thank you. This is not the right slide, Erica, right? So, but thank you very much, David, for the nice introduction. I'm going today to introduce you to a very powerful high-dimensional assay, which is designed specifically to decode inflammatory response directly from whole blood. And this is part of our mission to deliver translational relevant data with minimal sample processing as the open new possibility for immune monitoring in clinical setting. So next slide, please. But first, a little bit of background around inflammation. Inflammation plays a central role in many human diseases, from autoimmune disorder like rheumatoid arthritis to infection disease, and even in cancer, where it underpins immune-related adverse events during immunotherapy. While we know this for decades, our ability to accurately profile and understand inflammatory processes, especially at the cellular level, has remained limited, and most traditional methods measure the cytokine in isolation without linking them back to the immune cell that produce or respond to them. This disconnect is really a problem, and inflammation is not just about individual cytokines, it's about complex immune network where cells and signal interact dynamically in space and time. To make real progress in understanding the disease mechanism, optimizing treatment strategy, and identifying reliable biomarker, we need to develop tools that can capture this complexity. And that's exactly the gap that our team at Aliri set out to fill. A high-resolution, multi-parameter platform that reveals both cytokine expression and immune cell phenotype in a single integrated assay. Next slide, please. One of the major limitations in how we currently assess inflammation lies in the tools we use, particularly traditional cytokine assay. This method, like ELISA or multiplex bead assays, are excellent for quantifying soluble protein, but they give us no information about which cells are producing or responding to the cytokine. They also lack any phenotypic context. We don't know where the signal is coming from, whether it's from immune cells, stroma cells. This makes interpretation, especially in complex diseases like cancer or autoimmune disorder, incredibly limited. Yet we know that inflammation is not a simple linear process. It's shaped by dynamic networks of cytokine and interaction between multiple immune cell types. T cells, macrophage, dendritic cells, they all talk to each other, and that dialogue shapes the immune response. Without capturing that cell-cell context, we're essentially trying to understand the conversation by measuring just the background noise. And that's why we need a platform that can directly link cytokine to the cell that matter. Next slide, please. To unlock the full potential of precision medicine, we need to go beyond just measuring cytokines in bulk. Cytokines act as a critical messenger in immune signaling, but their impact is shaped by the cellular context which cells are producing them, which are responding. By linking cytokine profile to immune cell state, including activation status and phenotype, we gain a much deeper understanding of immune dynamics. This integrated approach allows us to decode the disease mechanism by identifying dysregulated immune circuits, monitor as well treatment response with more precision than soluble marker alone, and discover functional relevant biomarker that can guide therapy. In short, this level of immune profiling gives us actionable insights that are both crucial for research and clinical development. Next slide, please. So here, the introduction of the assay. The CyTOF-based panel assay allows us to simultaneously analyze 46 markers, including pro- and anti-inflammatory cytokine, chemokine receptor, cell lineage, and activation marker. What makes this platform unique is its ability to deliver a multi-dimensional immune readout, giving insight not just in cytokine level, but into which immune cell population are active, suppressed, or dysregulated. It enabled precise identification of immune signature linked to inflammation, immunosuppression, or immune escape with direct relevance for immuno-oncology, autoimmune, infection disease, and vaccine response evaluation. Importantly, this assay is ready to deploy, meaning it's available as an off-the-shelf solution that it's already validated on whole blood, making it ideal for both translational research and clinical cohort study. Next slide. Just an example of what this assay tells you what cytokines are being produced by which cell and what state those cells are in. You get full coverage from classical monocyte to cytotoxic T cell, all profiled at the single cell resolution in their native inflammatory context. And here, if you look, like at the graph, you will see for the T cells, you can really pinpoint, like within the T cell, the expression of a very specific interleukin. Next slide, please. So this is just a technology overview, and my colleague David is on the picture showcasing actually, like the CyTOF instrument. As you may know already, the CyTOF use metal label antibody and time of flight mass spectrometry detection. So basically, we are tagging the antibody that recognize all the antigen with a metal. And this has a lot of advantage over fluorescence because this allows us to multiplex beyond 50 markers with no need, for compensation because there is no overlay within, the spectral peak. Next slide. So this is a workflow that we have adopted to assess this particular assay in clinical setting. The workflow, it's intentionally very simple. The blood is drawn into PAX gene tube that allow us actually to stabilize the protein with a time frame of 72 hours at room temperature. That allow actually, the sample to be shipped directly to us from a clinical site. And everything, is shipped at room temperature. And after that, we start, the staining process by just, adding very small amount of blood between 250 microliter and 400 microliter into our labeled antibodies solution. We then, process to remove the red blood cell. We fix and we stain after that the cells for intracellular cytokine capturing the transient inflammatory signaling without labile manipulation. And once you can see, we have some stop in between that allow actually, to batch, the sample and to realize, for example, the intracellular staining just after a certain number of samples arrive, and we can analyze on the CyTOF all the sample from a large cohort. Next slide, please. So compared to the other method, our assay uniquely combine the whole blood compatibility with true single cell and intracytokine readout. And I put in purpose the other platform, like the flow cytometry platform, and as well as the LBA platform. Without naming Luminex platform, it's a platform that it's really used for cytokine evaluation. In terms of multiplexing, you can see that the CyTOF, it's over, 50 protein that can be analyzed at the same time. Now even more so with a barcoding, strategy. I can explain that, if there is any interest. Flow cytometry, in the best scenario, we are in the 10-20 protein range evaluation. And for the LBA, we're looking at about, 20 cytokine evaluation. Of course, the flow cytometry and the CyTOF have the cellular context that the LBA does not have. There is a whole blood compatibility with the CyTOF, which is also very limited for the flow cytometry. And also, realizable by using LBA. And finally, the sensitivity, and by what I told you about the ability of the metal labeling, which allow to have, a much more resolutive signal. The sensitivity of the CyTOF is higher than what you can get on the flow cytometry. Next slide, please. This is a case study where we were mapping inflammation in rheumatoid arthritis. The study context, we had the whole blood, and we performed the CyTOF profiling using the assays that I just introduced a little bit earlier in chronic inflammatory disease. We have some longitudinal sample at D0, D8, and D29 to look, like at the impact, like of the treatment after the baseline. We perform a direct measurement of intracellular cytokine and cell subcell cell, as like mentioned earlier. So using this whole blood CyTOF, we have observed in that particular case a coordinated reduction in pro-inflammatory cytokine, including the IL-6, the IL-1 beta, and then interferon gamma, and a compensatory rise in IL-10. This change were accompanied by a marked drop, as you can see in the graph, like on the right, in CD4 positive T cell, and as well as the monocyte. And overall, what we could say that this reflect a shift toward immune regulation for that particular case and treatment efficacy. And that confirmed that this assay has a lot of value as a pharmacodynamic tool in rheumatoid arthritis as well as other autoimmune disease. If you go to next slide, Erica. Finally, I just wanted to emphasize the translational impact of this assay. It goes, like really beyond research. It can inform patient stratification, help predict the toxicity immunotherapy, and support explanatory endpoint in trial. With real-world feasibility building, it's really ready for translational use in immuno-oncology program, autoimmunity, infection disease, as well as vaccine response profiling. Finally, next slide. To summarize, this is a takeaway message from this CyTOF assay. This assay provides an accurate, actionable readout of immune activation directly from whole blood. It's high-plex, high resolution, and as I said, already validated in whole blood, so it's totally field ready for clinical translation. And finally, next slide. So thank you for your attention, and I'm ready for the question.
Thank you so much for that, Corinne. Have a few questions here. So one of them is our current, the panel that we showed, our current non-proprietary panel, captures a whole lot of things. However, is it possible for us to add markers to the panel, or would we need to replace markers that are on the panel? And if you could give some idea of what the impact of that would be in terms of having it be non-proprietary or us running that.
It's totally, we can totally customize additional marker on this, like assay. So we can add, like marker on top of it. As I said, like the assay right now is limited, like at 46 marker, completely validated, but we can add on top of it. We have the ability without a multiplexing barcoding strategy to go up to 50 marker in that particular setting. So it's pretty easy, actually. We just need to customize a new tag because we have a lot of channel open for this particular platform. We just need to customize a new method for the new antibody and re-qualify that antibody within, like the large assay.
Okay, and there's another question which dovetails nicely with that first one, which is, are there any restrictions on what types of markers can be added? So what types of markers can be added to the panel, and does that depend on what's commercially available, or how do we approach adding markers to the panel?
So yeah, I mean, it really depends on what commercially available. We don't, like develop, like antibody against very specific marker at Alnylam. So we rely, like on what is commercially available. The whole, the whole customization for the CyTOF assay will start, like by looking at the specificity, obviously, like at the marker that we are introducing, the new marker that we're introducing. optimization of the signal, and qualification within, like the large cohort of marker, just to make sure that there is no interference.
Yeah, so Corinne, I guess for me to ask a little bit more around that, in terms of commercially available, we could also receive antibodies that have been provided by a sponsor, correct?
We could,
And I
we
guess,
have, and we
yeah,
do.
well, yeah, I just wanted to make that clear as well. We're not restricted to something
Yeah, we're
from
not restricted,
Abcam, et cetera.
actually.
The other
Everything.
question surrounding that is, do we need to use any type of secondary lab to do the conjugation, or do we support the conjugation of these in-house?
No, we do support the conjugation in-house. We process the whole, like as soon as we have the antibody, we will just tag the antibody with a metal, qualify it for specificity, signal to noise, and as well as steric inhibition when we are building the large, like panel. But yes, we do perform, like the labeling in-house.
Okay. And there's another one here in terms of how to use the assay. So, in our workflow, we showed our workflow using whole blood, but there are instances where people maybe have already done PBMC isolation, that they've previously collected and frozen. Are we able to do this assay on already isolated PBMCs?
We can, and we are, like in the midst of performing, like the validation of this type, like of sample with sample. But we have, like already good data that showcase that we can work directly on frozen, like PBMCs.
Okay. And I think that there's one more that we have, which is kind of at the heart of the matter is what is the deliverable? So, you've shown a lot of data, you showed, some case studies, but is there a report that's provided? A lot of people here, I think, are probably more familiar with flow cytometry, right, rather than mass cytometry. So how would this compare to a standard flow report? Is there anything that's similar or anything that's additional to what they maybe are used to seeing?
It's very similar, actually, like to a report from flow cytometry. Just, like the magnitude of the marker that we are using, it's obviously larger, but there is, like a gating strategy, which is always reported for flow cytometry. It's also reported for CyTOF. Just, like a reliability of the signal of the marker perform, like we just report it as well, like for cytometry, we report it as well, like on the CyTOF. We're making sure, that at least, like for each marker, we can comptabilize 100 event, meaning that we can see at least 100 cells. This is, like the base of having, like a good signal for every marker. And after that, we will perform, like the reading actually of those count and develop, like the graph showcasing, like the abundance of the different cell type, the abundance, like of the inflammatory profile, and some more, like deepening graph that showcase the in particular cell population, the cytokine level. But very similar actually to what you get, like from flow cytometry.
Okay, fantastic. And I guess I have a question myself as well, which is obviously we have an in-house data science team. How can we leverage our computational biologists on that team to help our sponsors design an appropriate, maybe experimental approach depending on what they want to see?
They will be able, like to understand what will be, like the variability of the staining, and they will be able, like to tell you exactly, like what number of sample will be, like accurate in order to get something, like reliable and can be, like translated into, like some clinics. They will actually as well, be able, and I showcase a little bit of a graph like this in a big cohort, of different sample to understand more, the disease mechanism. They will be able to perform, the clustering and the whole UMAP thing, and make sure, that every, heterogeneity within each sample, it's really covered through understanding of the different cluster, and some of them will have a translational impact.
Okay, and we just had another question come in. Can this assay identify whether some of the blood cells being activated during the assay process or after the treatment?
This assay can detect already, because it has a very high sensibility, can detect already, activation of cells without being, without being activated somehow.
Did that answer the question? All right. Does anyone have any further questions? If not, I would love to again take this opportunity to say thank you to Corinne for presenting this to us. As you guys saw from the picture, I'm very excited about this technology personally, based on my background. And so we really look forward to engaging with those of you who are interested. Our information's here. Obviously, feel free to reach out to us, and we would love to set up a meeting to discuss your specific needs and how we can help support your programs. Thank you so much.
Thank you.